course. This is a 2-day course spanning today and tomorrow. It's probably about five hours per day with breaks. And purpose of the course is to introduce you to some basic modeling techniques and to some software that we have that lets you build and run models fairly easily. So let me go to my slides. Let me just start.
Herbert, before you start, if I may, I just want to remind everybody, we are recording the session that we will post to our YouTube channel later, if you didn't see that note before. And I've posted in the chat the link to our Slack channel.
Oh, yeah, good point. Yeah. Yes, so if you look at
I didn't get that message.
look at chat. Yeah, there's nothing in chat now. Yeah, so we have a Slack channel if you're into Slack channels. We normally do this every year, and sometimes people use it, sometimes people don't. So it's available. It's good for asking questions in the background. There'll be people watching it, and they can answer any questions you have. Okay, let me share my screen then.
You can also ask questions in the chat here, and some of us can try to answer them, or we can just bring them to
Oh,
the
yeah.
speaker's attention. So
Yeah, that's right. Yeah, some years we get a lot of chat and less in Slack, and other years we get lots of Slack and less chat. So
yeah,
goes both ways. Okay, so welcome again. Yes, the pathway modeling course. Hopefully you can see my screen. Let me minimize that. So this year's 2 days. And the people involved are myself, Herbert Saro at UW. I'm a faculty in bioengineering. Lucian Smith, who's a senior researcher in my lab at UW in bioengineering as well. So this is in Seattle, west coast of the United States. And also Joe Hellerstein from eScience at UW as well, who's a faculty. His interest is, he has a lot of interest in modeling, but also recently a lot of interest in exploiting AI. So tomorrow we're going to have a special session on using AI in modeling. And then I also want to thank Hunter Robbins, who's the center. So this has been funded by the Center for Reproducible Biomedical Modeling. This is an NIH center. And Hunter Robbins is the chief administrator in that center, and he's helping us with all the logistics and the recordings and all the stuff that goes in the background that makes everything work. Okay. So oh, and here are the my partners in crime. This is what they look like. Okay, because my picture is obviously younger than I am now, but we'll keep that picture anyway. So that's myself, Lucian, Joe, and Hunter. So what are the objectives of the school? There's 2 days. Well, introduce some basic modeling into systems biology. This is really for learners, novices, and newcomers to the field. Learn how to use our Python modeling tool. This is called Tellurium. This is quite a popular tool. It's Python based, but we have other modes of using it as well. And I'll show 1 or 2 later today. So you'll learn how to do that. So it does require a little bit of Python, but not much. And I'll have a little crash course in Python anyway. But to be honest, you really don't need much Python to use this tool at all. And as I said, we have a brand new tool that we're going to roll out, I don't know, in a couple of months maybe, which I'll show you probably this afternoon or later this morning, where you don't need any Python at all. The other thing, we learn how to use biomodels, which is the big repository of models in Cambridge, Great Britain, and sbml and Antimony. And tomorrow we thought we'd spend a little time just learning the basics of model calibration and fitting. And something I didn't add here, which I should have done, is, in this day and age where AI is everywhere, whether it's ChatGPT, Claude, or Copilot, or Gemini, these tools can be used to help you with modeling, and we don't want to replace the human element, but they definitely help you and accelerate your workflow. if you use it appropriately. So we'll do a little bit of that tomorrow as well. We have quite a bit of material. What I'm going to do is I'm going to put this link in the chat. So this will take you, hopefully it'll work. This will take you to, get out of here. Come on. No, it's over here. It'll take you to the Google shared drive, which has a lot of material. And it has things like cheat sheets. There's a folder for the 2 days, and there's scripts, PowerPoints, and PDFs in the 2 folders. So you can use that material as you wish. It's there for you to keep. And then there's the Slack channel, of course, as well. If you're familiar with just Slack channels, you'd like to use Slack channels. Okay. So let me just, this is basically the 2 days. This is day 1. This is today. We'll see how we go on the time. The timing is always, rough, which is fine. That's okay, because it depends on, how fast I talk, whether I go off script, and whether there are questions from you yourselves. And we welcome questions. I welcome interruptions. I tell this to my students as well in class. Please interrupt me. I don't want this to be a monologue. And it's a lot more productive that way for everybody, because I get to find out what things you're not quite sure of or what things you're interested in. So it's useful for you to interrupt, okay? So this is the welcome bit. Probably going to be less than 20 minutes, right? But we'll dive straight into installing Tellurium. So we get the software sorted out. This is going to be pretty straightforward, actually. There are different ways to install Tellurium, but for today and tomorrow, we'll use a really easy one to do. And I'll also show you something you can use, other ways of installing it too. Then we're going to have an introduction to systems biology, and then we'll have a break, 10-minute break, and then we'll go into some practice and modeling. So there's a intro to Python. I'm not sure it'll take 30 minutes. An intro to the modeling language Antimony. Then we'll have a lunch break. Now you guys can decide whether you want 90 minutes or less. Okay, so 90 minutes is fine, but if you want only 60 minutes, let us know. Okay. And then we'll go into using Tellurium a lot more, and then also using some other tools. That's webiridium, not Iridium, webiridium, and then some advanced antimony work from Lucy. So that's today, and then tomorrow we'll have introduction to biomodels and general idea of reproducibility. We'll have some discussion time, and we may slot in more discussion times if you like. That's fine by me. And then some parameter fitting theory, and then some parameter fitting practice, more discussion time, and then the same lunch break, which can be short for or 90 minutes as you wish. And then we'll have a session on AI. Okay, so that should be fun. And the use of large language models, right? Not machine learning. The parameter fitting basically will use machine learning, but the AI will be large language model only. So that's basically the schedule, okay, for today. All right. Any questions so far? Let me stop sharing for a second. Any comments? Any questions? Okay, if you don't have any.
For those who have just joined, I wanted to remind we are recording today's session for YouTube, and someone brought up a point that they're having trouble accessing the Slack channel. It's working for some, I see, but if you have any questions that you need to post, feel free to put them in the chat, as Herbert mentioned previously.
Yeah.
Hunter, the link I posted, I went to the Slack and went to the invite other people and copied that link, and that seems to be working. So if you want to give that to the join.slack.com link that I posted on the general.
Do you want to post that into the chat and I'll copy it? And
yes, I did.
Fantastic.
I could put it in I could put it in
908.
Slack also if you want. Then it'll persist.
Put the Slack link in Slack.
I could put the Slack link in Slack because he's already in that Slack. And then you
Sounds
can
like
pull other people into the Slack.
That's not quite a chicken and an egg situation then. Okay.
That's right.
All right. Let's go straight in then to installing Tellurium. So I'll have a brief intro Tellurium is. Let me just share my screen so you can see something. And we'll go view. Okay. All right. So, okay, so the software. So there are a bunch of tools out there that you can use for, modeling and analyzing metabolism, gene networks, protein signaling networks. And what's nice about them is they're all compatible with this standard called sbml. We'll go into more detail later on that. I think Lucian will talk a bit more about that. But there's a standard called sbml, which stands for the Systems Biology Markup Language, which I'm sure some of you know already. That's used extensively in a number of fields. Those of you who do genome scale models, sbml is used always in representing genome scale models, and it's used a lot, at least half the time, if not more, in any other kind of modeling as well. And the advantage is that once you create your sbml model, you can freely move from tool to tool. You're not limited to just using, Tellurium. You can go to Copasi, for example, Pisces, VCell. And the key advantages, two advantages. One is tools of different feature sets. Okay, so you may want to use somebody else's feature set that another tool doesn't have. But what I've found, being an old person I am, is that tools tend to die off. They tend to disappear. Academic tools in particular, but also business tools, of course, also disappear. And when they disappear, you're sort of left on your own. And if you're not careful, you can no longer use the material you created with those tools. That's happened a lot. I mean, hundreds of tools have been written and most of them have disappeared. So if you relied on them, you're in trouble. But today it doesn't matter because you can just hop to the next tool that uses sbml and you can carry on going. Okay, so it doesn't matter which tool you use. But of course, we like Tellurium, of course, because we wrote it. But I think personally as well, it's a very high productivity tool. There was a review, last year in medium.com that went through about 15 tools, and they noted that Tellurium was a very high productivity tool, probably the most productive tool they tried. So, I mean, I'm a modeler myself, all right? I do write software, but I'm an actual modeler. And so when I model, I want to just, I have particular needs and I want to get on with the job. And so that ease of use is reflected in the design of Tellurium. The other thing about Tellurium is, of course, it uses Python, and these days Python will run on the desktop or the web. Now, I personally use it a lot on the desktop, of course, and I'll show you some of that. But very conveniently, it runs on the web too, and that's what we'll be doing in the next two days, because actually there's very little you have to do to install it when it runs on the web. So it's really easy for newcomers, but it's particularly easy if you're running a class. If you have a class of 50 to 100 students, or however many you have, it's a bit of a nightmare trying to install any software on everybody's, laptop or computer. You might end up spending the entire first class just getting everybody ready. But with the web, you just point them at the link, give them some brief instructions, and they're ready to go. So that's what we're going to do today. And what we're going to use is Google's Colab or Colaboratory. Okay. All right, so this is just, well, it's a bit of a busy slide, but the middle piece here shows the Tellurium software. So Tellurium is really an umbrella, and I'll show you another diagram in a minute. It's an umbrella for a lot of stuff. So Tellurium is just the top level of something that goes fairly deep down. But on the left, you see all these tools: Jupyter Notebooks, Colab, Spider, PyCharm, Basic, Python Idle, Anaconda, Visual Studio Code. These are all Python tools that you can use Tellurium with. Okay, so you can use any environment you want. Well, today we'll use Colab. The other thing, these are all these simulators down here, like VCell, Copasi, JWS Online, and Pisces, and so on. They're all compatible with sbml. So once you create a model with Tellurium, you can use it in any of these other ones and vice versa. So this gives you sort of a bird's eye view of where we are. This is more of a detailed map of some of the capabilities of Tellurium. So Tellurium has support for standards, modeling support, some utilities, and some differential equation support as well. And of course, you have all the MATLAB, sorry, not MATLAB, you have all the Python support as well. So if you use NumPy, Matplotlib, SciPy, SymPy, you know, any other tool that you like in Python, you can use those as well. Okay, so it's a great ecosystem to work in. And if you don't know Python and you want to be you want to be a computational biologist, I highly recommend you pick up at least a little bit of Python because it'll really help a lot with your work. Okay. So we'll be trying some of these. libRoadRunner is a key tool we'll be using. That's the backend simulator. This is a very specially designed simulator for high performance. It's very good at running in large clusters. It's very good as a backend for model calibration, where you're having to run hundreds of millions of simulations. And libRoadRunner was designed, actually it was designed originally for that use case because the bottleneck was always solving the differential equations in calibration. And the idea was to get that as fast as possible so you weren't limited in speed. So that's one of the prime use cases, but it's a general simulator. You can use it for all sorts of stuff. Okay, so Python. If you haven't used Python before, you may be thinking, well, where do I get Python from? Well, there's a site called python.org, okay? So that's nice and easy. But that'll give you only a primitive interface to Python. If you want to do real work with Python, you need to get a more sophisticated interface. So we'll talk a little bit about those. But even with raw Python, right? Maybe I can show you what raw Python looks like. Let me see if I can get Python up here. If I type Python, here we go. That's raw Python. That's raw Python. I assume you can see that. Big black screen with a little bit of text in the top. I don't know if you can see it, but I typed 2 + 3 and I got 5. It's very raw. There's full Python there, but it's limited in the user interface. For example, I can't do any plotting with this. Okay, so if I want to do plot graphs, I can't. But I can still run most of the packages, including Tellurium. And there's a very standard way of installing Python packages. It's called a pip install. And so if you ever use Python, you can use this command pip install Tellurium, and it'll take 30 seconds or so. We'll be doing that shortly with Colab. It'll install Tellurium. Okay. But as I said, if you just use the parameter interface, you won't get any plotting, right? And of course, plotting is half the fun. Okay, so you really do need to use something a bit more than the raw interface. And if you're absolutely lost, of course, in this day and age, 3 years ago, I couldn't have told you this, but if you're in real trouble, you can ask ChatGPT, you can ask it, how do I install the Python package Tellurium used in biochemical modeling? It seems to know a lot about Tellurium, and so you can use that as your little helper if you get stuck, okay? And, I use it a lot, of course. I mean, any of the AIs will be able to do this, but ChatGPT, Claude, and Gemini as well, but most of them will help you with Tellurium. Okay, so I mentioned fancy interfaces. Oops, that's not aligned. Fancy interfaces. These are a bit more than the raw interface, because if you start using the raw Python, you'll realize, oh, this is a bit 90s. So you need something a little bit more fancy, and there are a whole bunch of fancy interfaces you can get. I happen to use something called Spider. This is geared towards scientific computing. What I like is it's very interactive. Other people use PyCharm from JetBrains. A lot of people, my students, for example, will use Visual Studio Code. I have used it. I I just find it's too busy for my purposes. Because Visual Studio Code can, pretty much do anything. So, but if you like a programming environment, Visual Studio Code is the one to use. Jupyter Notebooks, of course, and we'll be using that through Colab. And then there's a new one which avoids a lot of sometimes the installation issues with these fancy interfaces, is Thorny. We discovered that a year or two back. It's a full setup for Python. You just install it, works on any platform, and it gives you the full setup. All you have to do then is use its user interface to ask it to install Tellurium, and it'll do it for you. So these are your choices. So as I said, we'll use Colab. So I'm going to copy this link into the chat because that's what you'll need. Okay. So we'll start that in shortly. As I said, this is great if you're a class or you can't be bothered installing something on your desktop or it's too difficult or whatever. This is a great way to get things started really fast. I just want to show you, this is what Spider looks like. I mean, I'm advertising this because I use it. It's sort of like a MATLAB interface. So if you use MATLAB, this is sort of similar. It has on the left-hand side, it has the editor panel for writing all your Python stuff. And then on the right-hand side, it has all the displays. It also has the interactive prompt and so on. So if you like that kind of interface, I highly recommend that one. So that's Spider. You just look up Spider, you'll find it. They keep updating it. It's now over at least it's version 5 point something now.
6.
Yeah, 6, really?
Yeah, it's 6. Anyway.
Okay. Last year was 5, so this year is 6. Okay. That's Spider. The other one, those of you who like programming environments, Visual Studio Code has become very popular with the developer community. It's a sort of shell that you can customize to do whatever you want almost. And so there are thousands probably of packages you can install to do almost anything. So you can use it to program in C, C++, Java, Rust, Julia, and of course Python. Okay, so and one of the great advantages of it is, unlike sort of like Spider, is it does source level debugging. So if you ever write very large programs in Python, this is a great option for you. The other one I should mention that does that as well is PyScripter, which is just Windows only, but that's also similar. But it's dedicated to Python, also has source level debugging. But if you write big programs, what's called source level debugging is really what you want, and Visual Studio Code will support that. For me, it's a bit too computer centric, so I don't tend to use it.
Spider will let you also do a certain level of source level debugging. I don't know if there's a significant difference between what Visual Studio Code does, but
oh yeah, no, this got proper stepping and breakpoints. Spider is done, everything's done manually.
You can like click in your breakpoints and stuff. I don't know, but
I think you have to then use command line to work it, unless they've changed it in version 6.
They may have like updates or something. But yeah, you also like the run button is different from the like run in debugging mode button. So, but yeah.
Yeah, but it has a command line debugger. It has the old GNU debugger.
Okay.
But it did. Maybe it doesn't anymore. Okay, notebooks. This is what we'll be using. So these are popular for various reasons. I think they're dangerous. I've seen so many people make mistakes with them. They rely on the idea that code exists in cells, these little notebook cells. And the other thing is that you can mix text. This is what people really like, I think, is you can mix text with the code. So you can always create like a book. So you can have all your text intermingled with code, and that code is live. Okay, so you can actually run it within the textbook. Yeah, my only issue is, and I've seen people make this mistake repeatedly, is you run something, you get some results, and you don't realize that the results are wrong because you forgot to run an earlier cell. So you have to be careful when you do that. But with enough discipline, it works. As I said, it's popular, so that's what we'll be using. Okay, so Colab, let's do this, shall we? Let's everybody see if you can install Tellurium and Colab. So what I'm going to do is I'm going to try and start from scratch. Oops. Oops, that's the wrong one. I'm going to try and start. I'm going to go. I'm taking a link, and I'm going to create a new page. Then I'm going to paste that link into my page. Now, because I've been using it a lot before, so there's a lot of stuff here already. But let me pretend this is completely new. So if you started off new, you might see something like this. So wait to make sure that people can do this. Right, so if everybody could try and do this. But watch the I'll watch the chat and Tony can watch the Slack. You should see something like this, okay? So this is running on a cloud. It can link with your Google Drive, which can be handy. It's free. Okay. There are paid versions. For the paid versions, I think you just get it's just faster, I think. Because in the free version, I think they you get to use their slowest computers probably. But I think you could pay, you get access to their much bigger computers, and you can just do bigger stuff on it. Okay. The other thing you'll discover tomorrow, Joe Hellerstein will discuss this. It's closely linked now with Google's Gemini. So you can actually, which is a very powerful combination, you can chat to your AI as you program. And that can be very, very helpful. Okay. Okay, so let's see if we can try it. Let's try this, shall we? So first of all, just make sure, excuse me, you can actually excuse me, you can actually run something. So hopefully you can see all this. I'm gonna kill the release notes. I've just this little rectangle here is called a cell, and in the cell you can write code. Okay, and if you want to create a new cell, you've got this plus code here. This plus text is the bit that lets you write just text, no program code, and this is what lets you pretty much write a book using Colab. So what I'm going to do here is what's up? I'm going to try and get it to do something. I've typed in here print 4 + 5. Right, hopefully that's self-explanatory. And I want to run this. Okay, now to run it, there's two ways. You can either do the easy one and just you see that big black arrow, click the black arrow, or you can do the less obvious way, which is shift return. I'll do both. I'll do shift return to begin with. You can see a little circle running round and round. It's starting up its Python environment, and it just did a program called Grammarly in the way. Go away, Grammarly. Quit Grammarly. There you go. And I have the answer 9. Okay. And just to show you something else, print hello class of 2025. That's a little message. And now I'll click the arrow instead, and I get the same thing. Okay, so you can, if you can't remember the shift return, you can just click on the arrow. Okay, all right. So what about installing Tellurium? Because Tellurium is not installed here. Okay, it has all the main packages, but Tellurium is one of them that's not installed. Now, as I said before, you have to do this. It's called a pip install, and I'm going to go back. I'm just going to go back to this original cell. Do it here. And to do that, I think you have to use the exclamation mark. It's basically an external command. It has to install it from the outside. So exclamation mark pip install Tellurium. Is it lowercase? I forgot if it's lowercase. Let me see. I think it is. Yeah, and I got to spell it right. So pip install Tellurium. Now it can be a bit chatty, and if you don't want it to be chatty, you know, talk a lot to you, you can do -Q, which means, you know, be quiet. It'll still say something, but it'll be a lot less chatty than it normally is. And then I can then just click the arrow and off it goes. So this will take a little bit. Here it comes. Oh, it's quicker than it. Oh, it's quite quick. So you should see something like that. Okay. So it's been installing various stuff related to Tellurium. So I'd like you to try this because you'll need this. You might get a message like that. Don't worry about that. That's something we'll deal with in a minute. So that's the installation done. Now, on Colab, however, you can't just run it. And the reason is because NumPy, so NumPy is one of the important packages. Yeah, good question. What is the one before the print? I have no idea. I tried to get rid of it, and it's
line numbers.
It's line numbers. Yeah, okay. Yeah, I couldn't get rid of it, I'm afraid.
Like if you have more than one line, it'll just say like 1, 2, 3, 4. That's the reason why it is there, is so that it can tell you like there's an error in line 3, and then you know where to look.
Yeah, there you go. It's a line number. There you go. The reason why it won't work immediately is because of this thing called NumPy. So NumPy is the big machine learning matrix array package for Python that you know, lots of people use. And Python has if I'm right, Lucian, Colab uses an old version, but we use a more modern version. Is that the reason?
Yeah, slightly. Yeah.
Yeah. So we use a more modern version. And so we have to tell it, it's okay. You can use the new version 2.3.1. And to do that, you have to go to the menu up here, look for runtime, and then ask it to restart the session and say yes. And when it does that, it should be ready to go, right?
Yeah. You can just import Tellurium if you want to check.
Yeah, so I'll just say it again. After this, after you see all this red text, just go to runtime and hit restart session, right? That basically restarts the session with now the new modern NumPy installed, okay? And it's ready to go. So to show you that it's ready to go, let me get rid of this stuff here. In order to use Tellurium and any package in Python, I have to import it. And to import it, you use the word import. Okay, now the other thing I'm going to do is Tellurium is quite a tricky word to type all the time, especially the R and the I often swap them by accident. So I'm going to recall it TE. Okay, so if you type import Tellurium as TE, so let me put these two into the chat. That's that one, and then this one goes in here. Okay, so those are the two you need, and then I'll run it. And look, nothing happened. That's a good thing. If nothing happens, that's a good thing. So actually, this means that Tellurium is actually ready to go. If I type TE dot, oops, if I type, let me do it again. I type TE dot, there you go. This little thing pops up telling you the kinds of things you can do with it, okay? Now, one of the things I'm going to do is I type get version info, right? So TE get version info. This is a function call, so I need to add the brackets and then run this. Now, I don't have to import Tellurium again, but it doesn't matter. I get the version number. So we're currently running Tellurium 2.2.11.1, and we're running RoadRunner 2.80. This is the ultimate test, I guess, that it's working okay. All right. So if you get to that point, you have successfully installed Tellurium. Okay, so it wasn't too painful, right? The only bit that's a bit of a nuisance is this runtime restart session. So if you install Tellurium again, if you do a pip install again, I think you have to run the restart session again, right? So tomorrow morning or this afternoon or another time you try to do this, and you will have to reinstall Tellurium every time because the session terminates and it forgets everything. You can save your files, all right, but the session itself terminates and you have to reinstall it, okay? But it's not a big deal to restore. But this applies to any package. So, but for Tellurium, you have the special thing of restarting the session, and then we're ready to go. Okay, so that's the installing. Let me go back to my slides. Okay, oops. So we've hopefully some of you have managed to do this. If you haven't, let Lucian or Joe let them know, or anybody else who's online that can do this. Okay, so you got Tellurium, and now you're thinking, well, now what do I do? Well, a couple of things. You can stay with us for the next 5 hours, and we'll show you lots of things about it. Or you can, oops, you can do this. You can go to our GitHub page. Let me go there. Let me put it into the chat. That's where we store everything on GitHub. And our GitHub page, this is it, Tellurium. All this stuff you can ignore at the beginning when it starts really here, okay? So it tells you what is it, gives you an introduction. And the important thing is the documentation, okay? So the documentation. So if I click on the documentation, it takes you to the documentation, okay? And it has installation notes here. It has a walkthrough. The documentation could be better. I mean, there's a lot of it, but it could be, you know, curated a bit better. So we're trying to get funding for this to do this.
But feedback is useful. Like if
if
you are trying to do something and you can't, like email us and say like, I tried to use the documentation to do this and I couldn't figure it out. So
I mean, currently the center helps with the maintenance, but for specific big projects like redoing the documentation, we're applying for additional funding to do that. So that's the documentation. As I said, there's quite a bit there, but it's getting a bit old, and maybe some examples need to be updated and so on. But there's a lot there. And as I said, actually today with today's AIs, you can just ask the AI. All right, so how do I do a simulation? How do I do this? How do I do that? And it'll tell you it because it's read all the documentation, so it actually knows anyway. Okay, so that oh yeah, if you're a Windows user, I actually personally recommend. Now, if you scroll down, you'll see there are installation instructions. There are various options here. One of them is the pip install, right? That's the standard one. But we have some specials for Windows. We have one special one for Windows, which basically it's a one-stop setup. It installs everything, including Spider and Jupyter Notebook interfaces. It's a no-fuss install, and we've got that down here, okay? So that's the one I use because I know it works on Windows.
I think it's up a little bit, actually, the IDE front end. But
no, this is, oh, is it down? No, this is it. That's,
oh, is that it?
That's it. Yeah, that's the setup.
Tellurium, okay, yeah.
Yeah, that's the one. Yeah.
Kiri actually just updated it, so you get the latest of everything too.
Oh, really? Oh, that's nice. Oh, nice of him. I added Thonny because as well. That's another one that's full install, no fuss. So that works quite well. We've got some instructions for the Mac. You can use the Thonny as well. You can also use Spider. And of course, you can do it on the Linux as well, and so on. And you can do it on Colab, as you say there. So you can install Tellurium pretty much anywhere you want. And if you do it on Colab, you can even use it in theory on your iPhone or Android phone. Not that I recommend it. It's a bit small. So, but you can certainly use it on a tablet, and I've used it on a tablet in classes. I mean, the touch control is a bit, getting used to, but it's possible. Yeah, so for Windows, there's a dedicated installer, but you can still install it on Windows using the traditional approach too. And for Mac and Windows, we have other ways. You can install things the way they install things too. There's the documentation, and I got a bunch of cheat sheets, so I'll look at that in a minute. So these are some of the things we'll look at today. Some basic stuff. We'll have an introduction to Python, getting the simulator running, which is you'll find out is really simple. And then building basic models, you'll also find that's really simple too. So we'll go through some of these basic things today. What else have we got? Oh yeah, the use of modelingai is a brief one-off slide. Basically, I tell my students this. Of course, many of us have access to AI resources. I mean, I must use it every day now. It's become part of a workflow. If there's some administrative thing I need to do, or some mundane work I need to do, or some advice on coding, I will tend to turn to these AIs for help. Sometimes they're useful, sometimes they're not. Often they're useful, but key thing about AIs, and I'm in my teaching mode here, is they will make mistakes quite often, actually, more often than you realize. So you need to be aware of what you're doing when you use them. Don't take their results blindly. So we coined a phrase the other day. So you've heard of prompting, and there's this new job called a prompt engineer. Well, there's another thing that we coined, which is post-prompting. Critically examining the results you got back from the AI. Okay. Excuse me. So we'll be doing this tomorrow, but have to watch out for mistakes from the AI. Okay, that's it. Okay. Any questions so far? No? Okay. Excuse me. So what I'm going to do now is I'm going to give a brief intro to systems biology. I give this set of slides to our students here at UW as an intro. Sometimes I wonder whether I should give it at the beginning or the end. But we'll do it at the beginning this time. So let's talk about what is systems biology. Some of you probably already know. Okay, so I won't spend too long on this. But anyway, what is systems biology? So, and here's an image from, this was generated, I generated this week. I did have an image before, but I didn't like it. And I made a new one. And this is the one I got from Copilot. I asked it, generate a picture that depicts the difference between the two types of systems biology, right? There's two types. There's what's called bottom up and top down. Bottom up has been around for a long time, and I'll show you probably one of the earliest papers on systems biology I know of. Top down is a new idea. It was born out of the new techniques that emerged in the late 90s, such as microarrays. And since then, of course, we have huge numbers of techniques that can produce large amounts of data, sort of under the title of omics. Transcriptomics, metabolomics, proteomics, any omics you like. Large amounts of data. And the task of top-down systems biology is to try to understand those large data sets. And because they're so large, it's very difficult to generate. It's very hard to generate a mechanism that explains this high throughput data. So most of they do generate models. Most of the models are statistical or machine learning-like models. Not always, but they generally are. And so that's basically the workflow for top down. One of the things they often do is try to do network inference. It's not the only thing, of course. And you often find these in the 2000s. It was very popular to draw these hairballs, with nodes and edges. They still do it today. I don't think they're particularly useful. But they'd get these big hairballs and say, look, that's what I've got. And then from that they try to distill out some hypotheses or some idea of what's going on. So that's top down, but the bottom up is what I do and a lot of other people doing, and it's much older than top down. So in bottom up, instead of starting high up with all the data, you start low down at the network level and try to build up. So you'll start with enzymes or genes, proteins, and from there you will construct bigger and bigger mechanistic models. Okay, so these are models that are built on biophysical principles. All right, they use chemistry and physics. They use chemical kinetics and enzyme kinetics, and they slowly build up these mechanistic models to try to explain some phenotype. And part of that is to build computational models, simulation models, from which people make predictions, and then they validate. Now the models have grown bigger and bigger. One of the biggest ones I know that's actually reproducible is about 80 enzymes. It's from a European group from Millard. Pedro Mendes was one of the authors. It's a nice model of E. coli. But there's a lot of groups in Europe, especially the Netherlands and Germany, who build these kinds of kinetic models. And very nice models they are too. So that's bottom up, and that's what we're going to be looking at. So so it's start with a single enzyme, add another enzyme, you've got two enzymes, add a third enzyme. Or if building a gene network, you start with one gene and you start another gene and so on. Or you're building a signaling network, you start with one protein and so on. And so you build up your mechanism, right? So there's some papers, there's been a discussion on, on the differences between these two approaches. They both have their pros and cons. For example, top down, it's very difficult to interpret the results because the scale is so big. On the other hand, bottom up is hard to scale up because everything is so detailed. There are pros and cons to both of these. But there's a paper here from 2013. It's a bit old now, but it talks about some of the lessons from top down and bottom up systems biology. This is basically what I'm summarizing some of the things I just said. So bottom up is basically hypothesis-based, model-driven, right? It's driven by models. And if you have a scientific model, you can falsify it. So that's one of the characteristics. They're mechanistic. They involve actual proteins and molecules. As a result, it gives you a better understanding and insight into how something works. And it relies heavily on theory, experiment, and computation, right? So there's a lot of theory as well. I mean, this diagram only shows simulation, but there's a lot of theory involved too. And there's generally a tight combination between experiment, computation, theory. Now for top down, it's sort of discovery based. You start with the data. So it is largely data driven. And you discover based on the data and the patterns you see in the data. And it's really the home of machine learning and AI. It's heavily empirical. It can use statistical methods. It's not very good at giving insight. It's generally theory-free, other than the use of some statistical theory. And then there's graph theory, but ultimately, I don't think the graph theory really helped that much. So that's sort of the differences between the two. So one is sort of more classical science, and the other one is more sort of a more modern scientific approach. There's another paper I'd recommend you read. There's a couple here. So I think it's from this paper, Biology Must Generate Ideas as Well as Data. So one of the criticisms of the top-down approach is because it's very data-heavy, it tends to be limited, as I said, in insight. That's primarily that it's limited. It tends to limit the insight you get into what's actually happening. And so there's a quote here, data should be a means to knowledge, not an end in themselves. Okay. So what's the first systems biology paper? This is just for fun. But I think the first systems biology paper is 1934 from Cyril Wright, who's a famous geneticist, United States geneticist. He's pretty famous in the genetics community. At that time, they didn't know, 1934, right? They didn't know about DNA. They didn't really even know about pathways. They had a vague idea maybe that there were pathways, that enzymes somehow made pathways, but they didn't know where the pathways came from. So there's this remarkable paper here that hypothesizes that, well, yes, there are pathways in cells. They're made from proteins, enzymes, and these pathways are determined by genes, even though at the time they had no idea what genes were. And so this is quite a remarkable paper that starts to think about cells as systems, right? And you can see some of these diagrams I've pulled out from the paper. You know, these are systems, right? These are pathways. That's probably one of the first papers. So what kinds of questions? These are the kinds of questions we don't ask in systems biology, right? So we try to contrast this with other kinds of biology. For example, we don't ask what is the gene responsible for coding a protein X. Now we need to know that, but it's not a specific question we ask. We ask somebody else. We ask, you know, a genomics person, what is the gene responsible for coding protein X? We don't try to answer that ourselves. Okay? We don't isolate the enzyme that catalyzes reaction Y. That's the realm of enzyme kinetics. We don't ask how some enzyme works, right? That's an interesting topic, but we don't specifically ask that question. We don't ask what the structure of a protein is. Right? Now, all these are useful to us, right? They feed into systems biology, but they're not the key questions we ask. We don't ask, you know, how does protein Z form a complex? You know, interesting, but we don't specifically ask that. And we don't ask things like, I need to find a drug that will bind protein Q. But we don't, that's protein structure, protein engineering question. Right? So the characteristic of all these statements is that they involve parts. Right? Enzymes, proteins, genes, they're all part questions. They're about the parts, okay? It's not about how parts interact. So if you look at sort of the pyramid, I can't remember what paper I got this from now, but if you look at the pyramid, you know, of life, so to speak, systems biology starts just above the parts. Okay, so you have the parts, genes, RNA, protein, metabolites. So we start where the pathways begin, and then it goes all the way up to tissues, and then to organs, and finally organisms. Okay, so some people call the top level systems physiology. Right, so that's where we are. So here are the kinds of questions we do ask in systems biology. Right now, you could just call systems biology physiology, in fact, because physiology is the study of functioning of living organisms, right, as whole things. Okay, so you could think of systems biology just as old-fashioned physiology. And so physiologists ask these kinds of questions. Why does the pathway use this feedback mechanism? What would be a suitable target for a given disease? Right, picking the right target within the system is the systems question. What computation, if any, do signaling networks carry out? Now, protein signaling networks, especially in mammalian cells, are very complex. They're obviously doing some kind of signal processing. What is that? Can we build a predictive model of a system, right? You're working for a company, you're trying to find a target, you want to know, you'd like to build a predictive model so you can predict the effect of manipulating different targets. You want to build a predictive model. How can I model something really complex like the immune system? That's a tough one. And that applies to any complex system. If you're a synthetic biologist, you'll be asking something like, how can I re-engineer this organism, right? Not the enzyme, but the organism to produce this valuable commodity. Okay. A lot of people, when they do metabolic engineering, focus on re-engineering a single enzyme, forgetting that that enzyme actually sits in a system and it'll be influenced by that system. So whenever you do metabolic engineering, you should be considering the whole thing, which is why, you know, people develop these genome scale models. And then it goes the ultimate question, right? What is life, the universe, and everything? I mean, that's really a systems question. You can't answer that by looking at just the parts, okay? But these are the kind of questions systems biologists ask. And so here's so you may be thinking, yeah, but isn't this just like biochemistry? Not really, not as far as I can remember. I did a degree in biochemistry, and it wasn't quite like systems biology. So biochemistry focused on the isolated parts. Now they did describe systems, but it was often verbally or as pictures. So if you look even today at any biochemistry, molecular biology textbook, it's stuffed full of pictures. Okay, we love pictures. So that's how I was taught biochemistry. There's really no theory other than descriptive theory. You may be thrown a little bit of enzyme kinetics, maybe, if you're lucky. But that's about it. And simulation, when I was taught biochemistry, our metabolic lecturer really did not like people who use computers. In fact, he had a term for them. He called them armchair biochemists. He didn't like them at all. He was a very classical biochemist where you go in, grind up, isolate, study, right? That was the idea. And any simulation you did sort of in private in your mind. So you did a lot of thought experiments. Okay. So the opposite, of course, to biochemistry, then we study intact systems, we describe systems quantitatively. We do use theory. For any of you who know metabolic control analysis or dynamical system theory, we do use theory. We use our math because that's a convenient reasoning tool. And of course, we use computer software a lot to do simulation. Now, you can still do thought experiments, and actually some thought experiments are really useful, but we do use simulation on a computer a lot. So these are the differences between biochemistry and systems biology. So yeah, I come from a biochemistry background, and I remember, you know, the classes were very descriptive. Zero theory. In fact, it was very much anti-quantitative, anti-theory sort of degree I did. So you may be thinking, well, maybe they had good reasons for this. Maybe because, you know, doing all this stuff on the right-hand side is a complete waste of time. Well, no. So I got a list here of success stories because the original, I suppose, is the Hodgkin-Huxley 1952 model of propagation of action potentials in axons, nerve axons. That's sort of the poster child. That was sort of the peak, actually. That was the peak. After that, it went out of fashion because you can see that the next paper I cite is 1973 from Katcher and Burns and Heineck and Rappaport, where they completely reassessed the operating principles of how metabolic systems operate. So that's a 20-year gap. I mean, stuff was published. It was largely ignored. And even this material, 1973, was ignored for about 10 years. And in fact, if we jump now 10 years before the next thing, I guess we got Goldbetter and Koshland on ultrasensitivity, the famous PNAS paper. That had a huge impact. Very quantitative analysis of phosphorylation, dephosphorylation cycles, and it turned out that they have special amplification properties, and they described it quantitatively. Then we're still jumping on, actually, 81, 97. Nice trypanosome model. This is sleeping sickness parasite. It's a prozoan. So they built a predictive model of trypanosoma energy metabolism and uncovered major constraints for drug targeting. Then we move on to, you know, the birth of synthetic biology with the repressor in 2000. And of course the bistable switch at that time as well. And then we have Bas Toesink from the Netherlands. He built probably the first really good predictive model of glycolysis in yeast. We have shortly afterward a credible model of amino acid biosynthesis. So amino acid biosynthesis is interesting because it's littered with negative feedback loops. And the question is, what are they doing there? And this one goes further on the biosynthetic pathways in amino acids. And this one focused on one path, pretty much one pathway with one negative feedback. The next one, 2009, looked at the pathways which had multiple negative feedback loops, nested feedback loops, and gives an explanation as to what are they doing. The short answer is they hypothesize that these feedback loops are basically balancing supply so that demand can always be met no matter what the rate of protein synthesis is. There's a nice paper in 2009 by Cole and Noble on the virtual heart. So we see sort of systems physiology. We have now the beginnings of iterations of models. We're not starting from scratch anymore. We're building on past work. This is due to the emergence of SBML, because SBML models are reusable. And so Smallborn has a nice iteration of 2.6 model in glycolysis. Jackie Snow, who developed the JWS Online tool, has a really nice malaria energy model, validated. And he's gone on to produce other great models too. What's interesting about these collection of models is that one of their approaches is they recharacterize the enzymes under physiological conditions first before they put them back into the model. And when they do that, they discover that the model just seems to work. And they don't have to do much parameter fitting. Okay, so okay, I've got up to 2015, but you can see, it hasn't been silent. There's been a lot of interesting stuff going on. A lot of this gives really interesting insight in how this stuff works, right? Which is one of the conditions of the human mind is I want to know how things work, right? It's built into us ever since, we left the trees. And this continues that tradition. So the other thing about systems biology is very cross-disciplinary. Okay, it covers everything. So when I did my biochemistry degree, the only extra thing I had to know was chemistry. All right, so we did a ton of chemistry. But today, if you want to do systems biology, you have to do a little bit more. Obviously, you need to know biology. You do need to know a reasonable amount of math, basic calculus, linear algebra, some stats. Control theory is very useful, not obligatory, but very handy to have. Dynamical systems is pretty important. And of course, models, building models, using some computational tool, be it Python, Julia, R, whatever it may be, whatever your favorite tool is. And the last thing is, it's rather important, being able to work with experimentalists. Now some systems biologists are joint, right? They're both experimentalists and modelers. But if you're not, you have to grow the skill on how to talk to the experimentalists, meaning what can they and cannot measure. Because you may have a very nice model, but things you need to validate it are just impractical for the experimentalist to get at. So it's always wise to talk to the experimentalist first, to ask them, well, what kinds of things can you measure? So I can work within those constraints, okay? So, and it also works the other direction. Probably one of the worst things an experimentalist can do to a modeler is, hello, modeler, I have all this data. Can we build a model? That's not going to go down well, because it'll turn out, well, what is your question? It's the wrong kind of data, et cetera, et cetera. So whenever you do systems biology, it has to be a joint effort between the experimentalist and the modeler. Otherwise, it doesn't really work. And that's a tough thing to achieve sometimes, right? Because most of molecular biology and biochemistry grew from my generation who did classes on biochemistry where quantitative studies were fairly alien. And there aren't that many experimentalists who can bridge that gap and talk to modelers. And also modelers, find it difficult to talk to experimentalists. So it's a skill one has to develop, but it's changing. So I mean, I talked to three experimental groups, and all three of them are really want to do modeling. So that's the perfect combination. Okay. All right, some homework for you. Not immediate homework, but for the next year or so, whenever you got time. I've got two papers here. You may know these papers. Well, you may know this first one. You may not know the second one. This is the Lasnik 2002 paper, published quite a while ago. These are thought-provoking reads. Can a biologist fix a radio? As a give you a spoiler, the answer is no. And the reason is because they focus on the wrong things. Essentially, the author says, and he has a point. So when they come to a radio, as we see on the left, on the right, is an old-fashioned radio, right? AM radio. They will start cataloging. I mean, I guess it's not surprising. I guess if you come to this, fresh, you know nothing about it, you'll start cataloging the shapes of things, the colors of things, the arrangements of things, how many things there are, and you'll end up with a vast catalog of components, capacitors, and so on, but you miss the whole point of the radio. So maybe there's a stage, though, where you have to do this first, right? You have to just get through it. And then you can start figuring out, well, okay, it turns out it really didn't matter that these capacitors were orange. What matters was it was connected to this black square thing here, and so on, right? So you have to then start building a theory of radio. And then you realize some data is not important at all. Some data is a lot more important than you realized. And some data you didn't even collect that you should have collected. So I guess, this is a natural progression, I guess, in any science where you first collect, then you start to rationalize. The other really interesting paper is this one, with Jonas and Cording, 2017. This one is, can a neuroscientist understand a microprocessor? So I grew up, in the 70s and 80s as a teenager, when the first microprocessors emerged, and it's a bit like AI today. It was like crazy time. It was, you mean I can have a computer in my bedroom, which was unheard of at the time. Now you had to build it yourself, but you could have a computer in the bedroom. And it was based on, the development of these things called microprocessors, which were basically a computer on a chip. They were relatively simple in those days. They were like, 4,000, 5,000 transistors in them. These days, they have billions and billions of transistors. But those days, you had, 4,000 transistors. And so the authors of this paper ask, if you're a neuroscientist, and I gave you this microprocessor, and this is an electron microscope picture of it, or microscope picture, could you understand what this thing is? And the short answer, again, as a spoiler alert, no. Again, it's because they have to start from scratch. They start measuring all sorts of things that are not relevant, some things that are relevant. They have no theory of operation. And that's the main problem. If you don't have any theory of operation, even if it's completely wrong, you really have no direction in which to collect data. And this is the difference, I think, between what's called data-driven and model-driven. So data-driven is purely collecting data and hoping that something will distill out of the data. Model-driven is where you have a model now, even if it's wrong, that gives you some idea, well, this, in order to falsify or disprove this model, I need to collect X, Y, Z data. Right now, in the process of collecting the data, you'll figure out that the model is not right or partially right, and then you can start the iteration of improving the model. Okay, it's a really fun read. This one, highly recommend it. Controversial to some degree, and one could argue, collecting data first is a natural progression, but at some point you have to break out of that and start formulating theories. You can focus your data collection more carefully. Okay, that's it. That's my slide deck. So, do we have any Any questions, or we can have a little break now, okay? Any questions? Queries? Criticisms? Or you don't believe anything I said? No, nothing on Slack either? That's okay. Okay, great, thanks. Yeah, no, I mean, I'm old enough to be able to say controversial things, so you know, these things have to be said sometimes. So and they may be true, they may not be true, but it, you know, thinking material, okay? So and those two papers are interesting to read. They've had a lot of citations, by the way, those papers. So people are obviously looking at them. So. Okay. If I go back to slide, let's have a quick 10-minute break. Give a chance to make a coffee or go, go to the bathroom or whatever. Because then we got an hour and a half of solid work to go through. And then we'll have the lunch break. Okay. All right. We'll be back at 25 past the hour, okay? 25 past, all right? That's a little bit 13 minutes. So I'll shut off my video and mute, okay? And take a break as well. Okay, see you at 25 past.